Thanks so much, Tim, and thanks, Cyprus, for that great introduction and very timely. We are a good-looking, undervalued healthcare sector company with a really interesting asset that is a great recession-proof concept for the next 12 months. So today, though, I'm going to talk a little bit about why this company is developing a really important asset for the world, for patients, and is a really interesting investment story right now. And very recently, we announced positive phase 2A clinical data with an endpoint called the CDR sum of boxes, which is the endpoint in Alzheimer's disease used by the larger companies like Biogen, who have already successfully had one antibody approved and are shortly about to have a second one approved using this same endpoint. So this is a very wonderful development for us, and it's our third clinical trial showing positive data. We do have a forward-looking disclaimer statement here, which I will not ask you to read in detail, but we will be making forward-looking statements as you would expect. So a little bit like Cyprus's chart, we've seen the highs and lows and excitement last year when the first Alzheimer's drug was approved in many years, followed by the crash in the market and a very strong rebound since June, where we've actually tripled in price, much better than Cyprus showed for the overall index as well. We are an Australian listed company. We have a major shareholder in the US called BVF Partners, which is a long crossover and venture fund, and myself as the second largest shareholder with nearly 4%. I've also put 500,000 of my own money in, not just, these are not just options as incentive payments. We have a very experienced chairman and board of directors. George Mawson, for example, used to be the chief medical officer at Amgen, a big biotech company when it was just in its early stages in the US, and a very experienced team. My own experience is most recently at a company in the US called Principia Biopharma, where I was the founding chief medical officer and took the company forward from a valuation of about 100 million to a final sale to Sanofi for 3.7 billion. The way we did that was to take two small molecule therapeutics through from preclinical all the way into phase 2 and phase 3, which is exactly what we're doing with our compound here at Actinogen called XanaMem. XanaMem is a once-a-day oral low-dose pill with a unique non-amyloid mechanism. And the reason I use the phrase non-amyloid is that the predominant theory for Alzheimer's disease is that the amyloid protein and tau protein that is laid down and is visible under the microscope is the main problem with the disease. But actually, it turns out that it's probably just a bystander effect, and that the inflammation and other processes leading to the deposition of that protein are actually the biology that we need to tackle. So our molecule does this by inhibiting an enzyme called 11-beta-HST1, which reduces the synthesis of toxic cortisol, which is a hormone the body needs to regulate cells. But you get too much of it as you age, particularly with Alzheimer's disease. And the drug has been shown to be rapidly cognitive enhancing, meaning it can help people think and focus more clearly immediately. You take the drug. It has the potential to be disease modifying, and I'll speak to that in a moment, and also a strong potential to be an antidepressant, which is why our second program is looking at depression. So we've treated already 300 people, including patients and healthy older volunteers, for up to 12 weeks. So we have a large safety database, and I'm not going to speak to safety today other than to say it's a very safe low-dose molecule at this stage. And we're moving quickly into the middle phases of testing in Alzheimer's disease and also in depression, focusing in depression on the foggy thinking associated with depression that commonly doesn't get better with normal antidepressants. And previous to the new study I'm just about to show you, there was a lot of evidence showing that this molecule works very well. So in animals, you can make a mouse model of Alzheimer's disease, and using a XanaMem-like molecule, we showed over 13 months that you can prevent the cognitive or ability to think decline independent of changing the amyloid plaque deposition. In other words, as the mechanism would suggest, this is an amyloid-independent mechanism. So it has very good preclinical or animal evidence. In humans, we did a human positron emission or PET scan showing that we have high levels of target engagement at very low doses of 5 milligrams and above. And this has never been shown with any molecules using this target before. So we are the unique human therapeutic that can test the so-called cortisol hypothesis. We see target engagement measured by hormones. One of those is called ACTH. And we did 2 separate placebo-controlled trials in older people who are cognitively normal, called XanaHES and XanaMIA, and both showed an improvement in attention and working memory within a few weeks of taking the drug. So a lot of support and preclinical and clinical evidence going into this program before we move on to the next study. So the study I want to highlight today is called our biomarker clinical study, and the design is shown here, and I won't dwell on too much of this, but previously we had a phase 2 trial of 185 people with mild Alzheimer's disease, which was very short, only 12 weeks, and didn't have a confirmation of Alzheimer's disease. And so it probably consisted of patients with Alzheimer's and a lot of patients without Alzheimer's. And not surprisingly, patients didn't deteriorate in the placebo group or in the active group during that trial, and it was overall a negative trial. But 72 patients had available stored blood samples that we could analyze to see whether or not they really did have Alzheimer's disease and were likely to progress during a shorter trial, such as 12 weeks. And the protein biomarkers we focused on were called p-tau, or phosphorylated tau protein, amyloid ratio, and some inflammatory markers such as GFAP. And we used a pre-specified analysis to reduce bias. And basically we chose patients with an elevated p-tau level who are likely to have Alzheimer's and progress, patients with a lower amyloid ratio for the same reason. And then we also focused on some other things as well in the analysis. The endpoints we were particularly interested in were the endpoints in the secondary endpoint list that are more sensitive in a mild patient population. And the one I'll focus on is called CDR sum of boxes. And this slide summarizes the key findings. So the major finding was that in this pre-specified endpoint of CDR sum of boxes, we saw a 60 to 80% relative reduction in progression of disease over 12 weeks. And in fact, basically, the XanaMIA group did not really deteriorate during the trial, but the placebo group did. So in other words, the molecule was protecting most people against progression. The effect size is fairly large, 0.6 to 0.8 points, and it's nearly twice as high as reported for an amyloid antibody, including the previous one called Aduhelm that did get approved by the FDA. And importantly, it wasn't just this functional endpoint that says how well you're doing with personal care and memory and things like that called CDR sum of boxes. There was another test called the test battery of executive function, which is more like a way of measuring people's ability to solve problems and think quickly. And that also showed a significant trend, as did another bedside test called mini mental state. So this was a very strong result for us. It showed that in patients within that study who had the real Alzheimer's disease and were likely to progress, you can see an effect of the drug quite quickly, much quicker than you can see it with an amyloid antibody, which typically is studied in trials of 18 months. So now our regulatory path to approval is quite clear and uncontroversial with this endpoint. And we have a really interesting data set to model our phase 2b study, which is going to commence in the next half, i.e. the first half of 2023. So in commenting on these results, Professor Michael Woodward in Melbourne said, these positive data for CDR sum of boxes and other endpoints are encouraging and indicate a likely therapeutic effect of the drug in patients. And the use of p-tau blood levels to confirm the diagnosis in future trials represents a practical and efficient method to find patients who are at risk of disease progression and in whom you can see a treatment effect. And this is very important for us because it means that we don't need to do expensive and complicated amyloid PET scans to get people into clinical trials. We can just do a simple blood test. And we showed in this study that it's highly effective for selecting the right patients to understand the effect of XanaMIA. And so at this stage, we're very focused on developing our phase 2b trial called XanaMIA. It will be conducted in patients with mild cognitive impairment and the early stages of Alzheimer's disease. It'll be nearly 330 patients, placebo-controlled. Patients will have a high blood tau level. We'll study 2 different doses over a 6-month period. And all patients in this study will go on to an open label extension study, meaning even if they get placebo for 6 months, they will switch over to active XanaMem at the end of the 6-month period. The endpoints will be multiple, but include the CDR sum of boxes and elements of the neurotest battery that we saw positive data in from the analysis. We'll be taking all of this planning and these new analyses to the FDA and the EMA in meetings over the next several months to get their agreement on the design of pivotal trials. Importantly, this XanaMIA phase 2b will be a pivotal trial that potentially could lead to approval of the drug in combination with other studies. We will be using our to-be-marketed formulation of tablets and basically designing this to be one of several pivotal studies to bring forward the day that we can get the drug approved for patients in Alzheimer's disease. And just a little comment on where Actinogen and XanaMem stand in the world of people trying to develop drugs for Alzheimer's disease. As I mentioned, the big companies have typically been focusing on amyloid approaches to the proteins, amyloid and tau, most of which have been unsuccessful or only just slightly successful. Nevertheless, the FDA is willing to give you a CDR sum of boxes approval with a 0.4 point approval improvement, and we are seeing improvements of at least 50% more than that. But compared to other drugs in development, which have a little bit of cognitive data, we believe we have the best cognitive data that has ever been published, because we now see both cognitive test battery data with a test called CogState and other endpoints like CDR sum of boxes and the neurotest battery. Most other drugs, including the small molecules on this slide, are actually developing an amyloid-related mechanism, whereas we have the only truly different non-amyloid mechanism based on cortisol. So we also have a major opportunity in depression, because we've already shown in healthy older volunteers that XanaMem improves cognition quickly, and foggy thinking is almost universal in poorly treated depression. Current antidepressants don't work very fast. They have a slower suicide risk in the first week or 2 of therapy. And there's a really good hypothesis that improving cortisol in the brain can treat depression and cognition. So that's the hope, that we may have a dual action to treat both elements of depression and cognition. And the study we have planned, which is starting in the next month or 2 here in Australia and in one selected overseas country, will be a simple proof of concept study over 6 weeks, where we add 10 milligrams of XanaMem or placebo on top of first or second line therapy for depression, which isn't working so well. The patients will have both depression and cognitive impairment, and the primary endpoint will be cognition, or the ability to think. And we will be studying the effects on depression and other aspects of cognitive function as secondary endpoints. The study is going to be relatively quick and efficient and relatively cheap to implement, because we can do most of it in Australia. And we hope to see results for this study towards the end of 2023 or very early in 2024. So our strategy at this point really hasn't changed with these new results, but it is really just validated and further strengthened. We're accelerating clinical development in these 2 diseases. We're forward planning for all of the necessary things to get the drug approved, such as good manufacturing practice, manufacturing and scale up of the tablets, getting ready for all of the different FDA and EMA applications you need to put in to get a drug approved. And then our focus is very much in the next few months on creating value from partnerships. We're actively engaged with a number of pharma and biotech companies that are interested in neuroscience, and particularly Alzheimer's. And we're very interested in evaluating potential regional deals, which could bring in some non-dilutive funding and perhaps expand the clinical capabilities of the partnership, and seeking value-add partnerships in various parts of the world. We're actively engaging with our regulators. We operate under a US IND for Alzheimer's, and we'll be having a number of important meetings and submissions over the next few months. And so just to finish off, we have a busy few years planned. This year, we're presenting at the San Francisco CTED Alzheimer's conference, where we'll be presenting the XanaMIA data from the April results of the previous clinical study. We'll be enrolling a trial in depression. Key regulatory submissions will be made, and we'll be starting to prepare the XanaMIA Alzheimer's trial. That trial will start enrolling in 2023. Partnering discussions are ongoing during this time, and we hope to have the depression trials results by the end of 2023, whereas the Alzheimer's study will definitely be a 2024 result at this point. And so with that, I'd like to thank you for your time.